Welcome class. Good to see that a majority of you attempted the quiz last week. On average, the class average was about 30 or on average 30 given the 43 marks. So good to see in them in terms of that. And most of the fundamental questions covered in unit 1 and unit 2. Majority 90% of you answered that well. But given some application oriented, bit more of advanced knowledge or application of the knowledge given from the fundamentals. Some of you are missed in that. So what I will do in this present class, we will revise a little bit more on the important questions missed in the quiz by majority of you. Take small concepts at a time, whatever we have covered, take in 1 or 2 slides. We will pick about why we came to the right answer. And then we will end up in the class to introduce a little bit to chapter 4, hypersensitivity and cytokine functions or interleukins. So that will be the plan for today's class. Tomorrow, day after tomorrow, we will go with hypersensitivity. There are 4 types of hypersensitivity reactions. We will see at the end of the class. So one of the question that hinge a little bit more on is this question. It talks about a clinical study is performed with subjects who developed sepsis. You know, right? Sepsis, when a body gets infection or a cut or a wound, there is some kind of bacterial growth happens in there. Eventually, it causes sepsis. Sepsis is no good for the system given the host. It need to be treated with, usually the bacteria cause the sepsis. It need to be treated with antibiotics so that you will kill the bacteria and make the tissue or the injury to come back normal. So in that study, they observed sepsis. They also observed that a wide-scale activation of macrophages in the septicemia or in the condition, enhanced condition of sepsis. They have seen a histological study. They have seen that in that site, more of the macrophages are there, particularly when blood clusters. And what are the pathogen in terms of sepsis causing pathogen? They were able to identify that a bacteria was observed. That a bacteria is of family Enterobacteriaceae. So in that study, clinical study, a bacteria was infecting some person or host causing a sepsis, and how the host is responding through the immune cells or defense, they were able to observe that macrophages are there, quite a high proportion of that. So which of the following substances is most likely to have the greatest effectiveness eliciting this macrophage activation? So they are saying that so what the bacteria is bringing as a foreign non-self molecule to the host that is suffering from sepsis, and then specifically what is the macrophage is recognizing from the things coming from the bacteria. And probably what we have studied that whatever is non-self, whatever is foreign, we termed it as antigen. Whatever that antigen recognizes, we termed it as antibodies. Antibodies are again given self and non-self. Given a non-specific and specific immune response, antibodies will come in only acquired immune response. But if you talk macrophages alone without involving any antibodies, then you are speaking about innate immunity. Macrophages are involved in innate immunity. They are involved in acquired immunity also as a crosstalk with the pathways. So basically, an example here they are talking. Yeah, any query? Someone was asking something. Any query there? This class you can stop and ask questions. It's a discussion class. Okay. If you don't understand anything, you can stop and ask again. We will get feedback from others also, your colleagues. It will be more of interactive session. Okay. Is it fine? Interactive session?
Yes, sir.
There? Okay. So what are they looking for? They are looking for the choices here. First thing, lysozyme, I can take it as it is coming from bacteria. Bacteria produces lysozymes to kill the foreign invading substances for itself. So I can eliminate this choice clearly. Lysozyme is part of the bacterial defense. So you are asking for which is eliciting from the host, coming from the bacteria. It is internal to bacteria. Lysozyme is taken off. Capsaicin is a different enzyme involved in different process. I can strike off this one. Thrombin involved in blood clotting. It's an enzyme protein complex. I can remove this. So I'm coming with complement 5A or lipopolysaccharide LPS. I'm coming with 2 molecules. And then if I look at the choice, what is asking, which of the following substances is most likely to have greatest effectiveness eliciting this macrophage activation? So basically, complement systems sometimes also enhance these macrophages. They may do it indirectly, but given a direct interaction with the bacteria, the system may be that to elicit the complement response, you need a first starting material, the foreign substance, that is antigen. So given these two choices, yes, complement may be there. It may be through antibody pathway, it may crosstalk with the macrophages, but complement 5A doesn't come without the antigen first. Without the bacteria or foreign antigen entering the system, complement pathway will not be activated. Antigen enters, antibody recognizes, antigen antibody complex, complement cascade comes. So this choice is a secondary response if you given acquired immune response here. But the first choice will be that the bacteria brings a specific foreign substance, that is a chemical molecule that's called lipopolysaccharide, that we already discussed in our classes. The first two response, innate and adaptive response. In innate, these kind of different these kind of cells are involved. In acquired adaptive immunity, these cells are involved. Antigen, antibody, this is specific, non-specific. Again, we have spoken in several classes that in innate immune response, one of the thing, direct response to pathogens is nothing but if a bacteria comes, it brings a substances called pathogen associated molecular patterns, PAMs, that we already discussed. This may be lipopolysaccharide coming from bacteria recognized by toll-like receptor or directly from macrophages. So the innate immune response, maybe antibody or acquired immunity may not be involved in this system because too much sepsis is happening. So there should be a quick response to kill the bacteria. So the body might prefer to go with the innate immune response rather than adaptive immune response. So lipopolysaccharide coming from bacteria recognized by macrophages, they will respond specific macrophage response such as inflammation or cytokine production, that is here. So the answer for this study is lipopolysaccharide, that is the first non-foreign molecule. But in given a scenario, if it is acquired, you may pick the complement if lipopolysaccharide is not given. This choice removed, these four choices are given. I might prefer to pick complement based on the acquired immune response. So that's a question one. So it basically introduces how your system defends fundamentally. What is the antigen coming from foreign bodies, foreign entities? So, okay, the second question again related to that, a major factor regulating adaptive immune response is. Again, you are talking about acquired and adaptive and innate. In the adaptive immune response, what is the major factor you are speaking? Again, if you talk in terms, haptoglobin, we don't know. We haven't talked about it in the class. Neutrophil, we know something about it. It may do something in terms of phagocytosis. But is it part of the adaptive response immediately molecule helping in the adaptive response? No. Actually, adaptive, if you talk humoral cell mediated, humoral by antibodies, cell mediated by T cells. Then these two molecules maybe go in the end, talk to the neutrophils to damage specific person, specific foreign molecule. But the major factor given the system here, neutrophil is not, haptoglobin is not. If you take complement system, yes, it may be a major factor antibody-antigen interaction. But again, as discussed in the previous slide, previous question, it is complement is a consequence of antigen coming and binding to the antibody. Without this antigen coming in the system, complement system may not be enhanced. It will be in a dormant stage. So given the same concept, fundamentally, the concept comes into that antigen concentration plays a crucial role. And again, we spoken, same thing, antigen, antibody, antigen comes here. These cells will come.
Ooh la la! Ooh la la!
Secondary response. Okay, so we need to go into the participant and mute all. If you have any questions, you just put it into the chat box. Okay. Okay, that gives back. That comes back to that. You need to fundamentally work on these two. What are these cells? How they work? You have to make notes of it. Then you go into the antibodies and antigen, how the interaction we already spoken. Antigen brings specific chemical entities. They go in the recognition process, go into and study the fundamentals. And we specifically spoken in terms of generation of antibody diversity or acquired immunity. After repeated stimulation by antigen, B cells can make antibodies that bind their antigen with higher affinity. So you're talking about a stimulation by antigen, not only one time, a repeated stimulation, primary response, secondary response. Again, the concentration of the antigen is also important. How much antigen is available? If only few molecules, they may not elicit the response. If given a proportion, they may respond. So the answer for this is again antigen concentration. These two choices will not fit in the answer, but complement can fit given that secondary response after antigen antibody interaction. So again, go and look into the cells. What are the cells involved in that? We may pick some of these cells in further classes, and then mainly neutrophils, basophils, mast cells. These are all involved in allergic reaction involving same mechanism, again humoral and cell mediated only. But many of the cells, given scenario, they act in differently. So in terms of lymphocyte versus phagocytes, you should also look in terms of the what are the phagocytes, basically innate immune responses. What are the lymphocytes? T and B lymphocytes, mediated adaptive, mediated innate, but crosstalk might be always there. The tissue specificity will be here, tissue non-specific will there. Most of these, what are the phagocytes? We will look in further slides because there were few questions in that. So coming on to now complement system, several of the complement components are. So we are asking a specific question on the complement pathway and what are the components involved in the complement pathway, and then what are the in terms of fundamental nature of these components. You have several of these here. You have cytokines. Do you think complement components are cytokines? The choice is that cytokines basically come from T cells. T cell cell mediated immunity. They produce cytokines after specific recognition through MSC molecule of specific antigen. So complement involves antigen antibody binding coming after that classical pathway or two other pathways. So basically cytokines coming from T cell, a complement system, it is not involved. And then antibodies, let's say antibodies, you can think of this, we will come back. Hormones, do you think hormones? They are not hormonal response, primary and secondary messenger. This choice is gone. Glycolipids, it may be possible, but we have seen already that most of the antibodies or antigens are glycoproteins. Few of them are glycolipids. So it may not be a feasible choice here. So given that antibodies and enzymes and complement components are again, it's a secondary response of antigen antibody reaction. So they are coming after antibody antigen interaction. So this choice goes again. It's part of the antibody antigen complex complement pathway, but they are not the components. Major components are, we've spoken that how a cascade, enzymatic cascade works in the class. Enzymatic cascade in the factor C, which is in inactive stage, activated. Factor B, another activation of enzyme 2 happens, complex goes, and there is a complex cascade. Clotting enzyme becomes clotting enzyme. We have discussed on the enzymatic cascade and how enzymes work, and we have spoken that the complement system consists of these C1 to C9 class of plasma proteins, and we have discussed that they are nothing but they are a kind of enzyme complexes. They go from inactive to active state. Why they should be in inactive state at the first place? Because if they are active, you are going to destroy your own system. They are going to perforate their own cells of the host. They don't need to do that. This activation happens through a specific antigen comes foreign. Antigen antibody interaction comes. Then the inactive becomes active. Cascade comes and produces the C9 complex that destroys the pathogen. So given the scenario. So they are nothing but they are enzymes. Several of the complement components are called C1 to C9 complement proteins, plasma proteins. They are basically enzymes in nature. This is very fundamental question. You should look a little bit more deep into that. So these three choices you can delete. You might have most of them are picked this choice, but the preference choice is enzymes. Okay, another question in the other question, a study is conducted of immune responses to antigen. Okay, antigen is coming to the body. What the immune responses a host elicits, it is shown that immune responses to every type of infection encountered by T cells and B cells. They're specifically showing that T and B cells are involved. They're talking about acquired immunity here. And they're saying that through which of the following mechanism This the diversity response most likely to be developed. They are saying that excuse me, there are billions and millions of antigens, foreign molecules coming into the body. The same way your immune system responds by producing millions and trillions of B cells or T cells, depending on the antigen, depending on the immune response, a type of the immune response involved, humoral cell mediated. They're asking that how it is generated, how this diversity is generated. The choice is expression of different CD receptor, MHC class 1, class 2, selection in thymus, or encounters with infectious agents, chance genetic recombination in utero. Now you see in that in all the 5 choices, the question is asking like what is the mechanism? What is the mechanism involved in the diversity? These other choices may be given here. They may be a kind of prerequisite step to start the mechanism to go on. But fundamentally, how do you generate a same B cell to produce different antibodies of different specificity? The closest choice here, you go look into that. We have spoken in the class, molecular basis of generation of antibody diversity. Several mechanisms are there, multiple germline gene segments, combinatorial VDJ joining, somatic hypermutation, that also we said, yeah, hypermutation is involved. So if you think of the mechanism, a recombination happening here, only one choice that talks about any of these mutations here. Nothing talks about the mutation. Chance genetic recombinations in utero during development. What happens in the development? So a kind of mutation is involved that we already spoken, a kind of gene rearrangements given a molecule constant. They rearrange in different times before either splicing happens also. These molecules can sometimes rearrange themselves given the, what do you call as germline development, somatic development. It's a bit fundamental. We will see in a given, we will expand the concept, but it's a random and gene rearrangements. That is nothing but genetic recombination is the mechanism. If they talk, what is the first response? They are talking T and B cells on both. So when they talk both, T cell only involves CD4 and then MHC molecule. B cell doesn't involve them. So when they're talking both cells, these two choices can go away. And thymus and bone marrow, yes, it's fine. Thymus and bone marrow, they encounter them, but that is the starting encounter point where the lymphoid organs are there. But this is not the response you're looking for. Encounter with infectious agents, again, it's a prerequisite primary response. By doing encountering infectious agents in the thymus and bone marrow probably B cell, thymus T cells, they may elicit an immune response. They will generate the diversity through going with this mechanism of genetic recombination. So that is the choice. It's fundamentally very important. We will see that in the coming classes, how T cell diversity can be generated. That we will see in unit 5, I think so. Again, same VDJ recombination. You have given 40 V units, 30 D units, 6 J units. What is the possible variable regions of antigen receptor molecule you are looking for? So either it's 40, only V regions, 76, a combination of all these three in a kind of addition, 40, 70, 76, or is it a multiplication, 40 into 30 into 6? So we have spoken that VDJ recombination given estimated number of segments. What are that? Estimated combination of that. You can simplify, you can see into that. You are multiplying the rearrangement, recombination, given a probability, a given scenario, you can generate maximum given this variability. Same way here, 40 into 30 into 6, the answer is 7,200. It's again fundamental of the recombination, VDJ recombination process. So one more question, it's talking about again on VDJ, a recombination of VDJ or immunoglobulin gene segments. So they are giving specific choices saying that it only occurs in mature B cells, only occurs in light chains. It involves heptamer, spacer, heptamer flanking sequences. It is affected by recombinase enzymes. So these first two we already discussed. It's nothing but a recombination will happen both in light and heavy chains. Both light and heavy chains can give a new recombination. For example, you're talking about heavy chain here, but light chain also can give recombination changes, factors. These factors you need to multiply again. Given the 8,000 here, taking only heavy chain, it can recombine, it can go into permutations in many combinations. Given a scenario, if you bring both heavy and light chains, you may generate 2.6 into 10 power 6 variants of antibodies in given scenario. So it involves both light chains and heavy chains. It doesn't occur in mature B cells. We have spoken the concept in the class. It starts from pro-B, pre-B and mature B, mature and peripheral B cells. So there is a process starting from in utero development, progressively goes into that. So it involves both immature and mature B cells. In immature B cells, there is recombination might happen. In mature B cells, splicing can happen maybe. So it involves heptamer, spacer, heptamer flanking sequence. Yes, we have known that. We have seen some sequence similar to that. But whether it's heptamer, or it's something else, we will go back to the slide. And we coming on to choice 4, it is affected by recombinase enzyme, RAG1, RAG2. This seems to be closer answer for me because we have spoken in the class. We have spoken that there is a recombination signal sequences, recombination activating genes or enzymes are involved. So which of the sequence correct? Choice 3 or choice 4? We have to come back here. So we have spoken different segments, heavy and light chains are involved. We have seen the RSS sequence. It is not heptamer and heptamer. It is heptamer, spacer and nonamer. So 7 and 9. So basically choice 3 is taken off. This is not the correct choice. These two are not correct. So we are going into specifically, yes, the mechanism of cutting and pasting, the specific splicing happens. It is involved by RAG1 and RAG2. That is the main excision happens here, loop excision, VJ recombination happens here. So the answer is answer 4, that involves mainly the correct choice. This is close, but it should be a nonamer here. And these two are fundamentally incorrect. Okay, next move on to a little bit more on lymphoid organs. Where are the lymphoid? In a given a body, given immune system or immune cells, we know that in our body, we have immune cells working 24/7. They may not be sitting at their origin. For example, T cell thymus, B cell bone marrow. They don't be sitting at the origin. They may be moving into primary to peripheral lymphoid organs. They may be going into lymph nodes. They may be going through blood. Their job is to surveillance, right? Any foreign molecule comes up, they will take it up. So this question is asking about upon taking the shortcut between sidewalks, a student walks across the lawn on campus and stops briefly to reposition his backpack. He looks down and realizes he is standing on a fire ant mound. So saying that suddenly he comes out of the pavement and he steps on a grass. There is some kind of ants colony are there. He stepped on the ants. One ant bit him. And then 4 days later, he could see that lymph nodes are enlarged. Groin region means near the neck, right? So which of the vascular tissues is most likely to play the greatest role in leading to this immune response to the bite? He's asking. Basically, a human body, for example, a student has a proper immune system in working place, and he encountered a foreign molecule or foreign toxin from ant bite. Given a system, he could see that lymph nodes are enlarged. They are asking that where the immune system is probably responsible cells are floating in his body. So given here, you have lymphatics only, it's related to lymphatic system, lymph nodes, leaving all the 4 are blood vascular system only, veins, capillaries, arterial, arteries. As a common sense factor, it is a blood vascular system, anything can be involved during the blood flow. Veins or arteries carry different types of the blood. So I can basically delete all the 4 choices. If given scenario, given that any immune cell is flowing, we know that it flows through the blood or circulatory system. We never spoken about that it flows through only capillaries or veins or arterioles. So it goes through circulatory system, it involves everything. So the right choice here, the lymph node involves lymphatics or lymphatic system, either peripheral or primary, peripheral or primary immune lymphatic system. So the closest choice based on common sense, I will pick the lymphatics. That we already seen here, primary bone marrow, fetal liver, where the B cells are here, thymus, T cell. They go to different secondary lymphoid organs. Most importantly, you should be very careful or very fundamentally should be strong in terms of when the antigen enters, for example, transplantation rejection also, when the antigen enters from the foreign graft, it will be available only in the secondary lymphoid organs. They may not go to the primary lymphoid organs. So these secondary lymphoid organs, per se, lymph nodes, are the major site where the cross communication between self and non-self happens. Foreign antigens come there only, circulating immune cells will reach there, and along with the immune cells, these innate immune cells, for example, natural killer cell, monocytes, platelets, all of the mast cells, they may be in the lymph node also. The lymph node antigen presenting cell will give the foreign. Lymphocytes are there, they take a proper step. They need assistance from macrophages or dendritic cell to induce phagocytosis. So all of them are in one shot, just like breakfast here. So you have breakfast, brunch, lunch, everything here. So given the system to work properly, they will immediately work through, kill the foreign molecule here. So this is important. You need to know that where the cells are circulating, where they are eliciting their immune response, in which cells. Mainly, the lymph nodes are really important. So the answer is lymphatics here. The next question is also same. It is whatever I explained in the previous slide. They are saying that T lymphocytes were there. Where is the proper tissue or action can take place in the given antigen of that? So where is the tissue or action taking place? It mainly takes place in the secondary lymphoid tissues. Again, the origin of the immune cells, much of the response does not happen, whereas other the peripheral tissues are important. If given scenario, the answer here, basically thymic medulla, bone marrow, these two are the primary lymphoid organs. Spleen also primary lymphoid. Mucosa may be a kind of interstitial intermediate, but given the scenario where much of the action happens in immune system, I will pick the choice of gastrointestinal mucosa or lymph node paracortex. Given these two scenario, I will basically pick the choice lymph node paracortex given the scenario of secondary lymphoid tissues. So that is the answer for this. So you can go into that. This slide is also important. Zoom into the immune cells. Look in the body as a human being whole, where as a whole system, holistic development of immune response. Look into the primary, secondary. Go zoom into the lymph node here. You see lymphatic vessel, blood capillary. You may see a lot of macrophages, antigen, antibody, a lot of like it's kind of a chimera here. Not chimera, it's maybe multiple. Lot of complex foreign, non-foreign self and macrophages, innate cells, all will be acting in the lymph node. That's why when you get a fever, first the doctor checks that do you have any lymph node inflammation, saying that you are really fighting for the fever, some bacteria and viral fever. That they will test that. If lymph nodes are inflamed, then you need to go for antibiotic course, given the bacteria, other system involved. So the answer is lymph node paracortex again here. So again, depending on the immune cells involved, this is also important, phagocytosis, what type of immune cells are involved. Given scenario, only one thing is odd man out. We know that natural killer cells do phagocytosis, macrophages do phagocytosis. We already know that because they are part of the antigen presenting cells to the T cell mediated immunity. Neutrophils are also involved. Given the neutrophils and basophils, which is the best choice? You go and look into that. What are the cells? Phagocytes, basically dendritic cell, macrophage, and neutrophil. And natural killer cell, it's we will see that why they will be involved in phagocytosis. Because natural killer cell, they may not directly phagocytose, but they may provide some molecules called cytokine production, interferons alpha and gamma. One of the interferon coming from natural killer cell may induce some of these dendritic cell or monocytes to go into the phagocytosis mode. It may not be directly involved, but it is indirectly involved. But given the scenario of macrophage, dendritic cell, neutrophils, they are directly involved in phagocytosis. Natural killer cell or indirect response. And given the basophil, this is not involved in phagocytosis, but it is nothing but it has a molecules loaded with chemical molecules called histamines or interleukins. When the basophil encounters an antigen, it does want to give a response, immune response. It goes into a diffusion. It releases all these molecules. Just it bursts out. It releases histamines. It releases interleukins. They will take a specific action. The increased vascular permeability means allowing the blood vessels to dilate so that more immune cells can come up. This is a involved in the allergic reaction, allergic asthma. Basophils are involved. That is chapter 4. We will look into that. So given the choice, these two are directly involved, indirectly, natural kill indirectly involved, macrophage, neutrophil directly involved, given that basophils are not involved. So this is the choice, odd man out. So again, protection against microorganism inside cells or peptides derived from intracellular infectious agents. So the choice is either T cell, B cell only, the first responder. This is the secondary consequence. All three can be taken out. Whether T cell or B cells are involved, we looked into that when intracellular pathogen, antigen coming from digested pathogen. This slide is very important. It talks much about the cell mediated fundamentals. In a given time, you need to make notes of it. If a pathogen is coming inside the body, taken out into the dendritic cell here, digested, antigens are coming up, giving to the T cell. So only antibodies directly bind to the humoral in free floating, the soluble antigens. But if not soluble, it's inside the microorganism. You need to talk about T cell mediated immune response. So the answer is T cells here, not the antibody. If antibody goes out, all these complement system is due to antibody only. All four choices goes out. So complementarity determining regions. This is fundamental again. We looked in the class that different parts of the antibody, FC we spoke, FAB here, light and heavy chain, variable region. We have spoken that there is a specific region here in the variable region where there is a specific complementarity happens. So variable regions are sometimes called complementarity determining regions because they know how to bind with the antigen, complementing with the antigen specifically. How they will do that, we know about it, that there is a kind of spatial complementarity. There is a hydrophobicity, different weak bonds are there. Then complementarity happens here. Those variable regions are called complementarity determining regions. That's why CDRs is nothing but the complementarity is concerned with antigen recognition. So how they will do, this is the thing we have spoken on. Again, not only B cell, T cells are also MHC2 molecule also. We have spoken tertiary complex, a alpha and beta groove comes. There is a cleft, that cleft also nothing but a complementarity determining region. I think most of you answered these two questions wrong. Look into the fundamentals. This is also CDR is nothing but complementarity determining region. So intracellular pathogens, parasites again killed by, when you talk about parasites, you talk about T cells, mainly that gamma interferons are here. We have spoken, we have less than 1 minute, so I will go quick here. Gamma interferon is the answer. Aptan, chemical molecule, we have spoken, not the choice one, choice two here. Look into that. And then immune responses are depressed by mainly stress. You need to have a very healthy body, healthy system, so that you have a proper control over sleep and diet. Immune system works well. If you are stressed,